Hello, and welcome to the lecture on T cells. So we're done talking about innate immune cells, and now we're moving on to the adaptive or long-term immune cells. So one of the most important concepts in all of immunology is happening here. What are the differences between, I've said this a few times, MHC, that stands for major histocompatibility complex 1 and 2. That's a good part of today's lecture. The other part is what are the differences between different types of T cells? They're most often called CD4 or CD8 T cells. And then we're going to go back to the thymus. So one of the earlier lectures went over the primary and secondary immune organs. Primary are where immune cells grow up. So it's definitely the bone, but the thymus is also considered primary because that's where T cells become educated. And we'll talk about what that means. And then lastly, we'll talk about what would happen if you did not have T cells. All right, here are some keywords. We have mentioned antigen presentation before, particularly with innate immune cells that are good at eating things. So phagocytosis. The cells that are best at eating things are macrophages and dendritic cells. And yeah, we'll go over that. And once we get to the brain, you'll find out that there are antigen-presenting cells in the brain too. They're called microglia and astrocytes. Okay. So how on earth do you identify self from non-self? This is an essential concept for people who are preparing to receive a transplant or, doctors and nurses that work in the transplant unit. All right. So how do we identify pathogens from our cells? You know this. It's Toll-like receptors. But there's more to it than that. Like, why can you not just get blood from somebody else or somebody else's liver? Like, why can't we do that? Because of receptors called major-histocompatibility, meaning compatible with tissue. Histo almost always means tissue. And that's on the surface of just about all of our cells. There is an exception. I think I'll wait till we get there. Selecting for T cells which properly respond to these receptors is the job of the thymus. And then transporting and identifying antigens in the lymph system is the job for the innate immune system. So we're going to talk about where they intersect today. All right, so here's like the simplest drawing of an MHC class 1 and an MHC class 2. I will tell you more. MHC class 1 only has one anchor into the cell membrane. MHC class 2 has two anchors, so it's actually easy to remember their names based on their shape. Now, where they get these little red circles from are very, very different. That's a core concept. We're going to go over that several times. Okay, so everybody's major histocompatibility complex is a little bit different because they're made up of 200 genes. However, we fall into like groups. So there are 5 to 6 different features that people in transplant biology look for. And like a 6 out of 6 is a perfect transplant. 5 out of 6 is fine. But most of the time they wouldn't do 4 out of 6, definitely not 2 or 3 out of 6, because that person's immune system would reject the tissue. Okay, so it's present on almost every cell of the body except for red blood cells, because red blood cells actually kick out their nucleus. So the part where the DNA is contained, they kick it out. 2 reasons, mostly because they don't need it and they're not going to live a long time, just a couple of weeks. And the other reason is to make room for more hemoglobin and oxygen. Okay, so it allows for an infected cell to communicate with other immune cells. So let's say, there's going to be tons of examples, so don't worry. Let's say that a lung cell got infected with the influenza virus. How can it tell? How can it tell a T cell? So there's a step-by-step drawing for that. But what they do is they take part of the virus break it down and put it on an MHC class 1 on the outside of the cell. And if it's on the outside of the cell, CD8 T cells can come and find it. Natural killer T cells can come and find it. And this is the smaller one. It presents only 8 to 10 amino acids, and it's usually derived from the cytosol. This is important. I'll tell you why. Because MHC class 2 gets its proteins from a different spot. So cytosol is just like the fluid inside of the cell. Here we go. All right, so here's your virus. It's infected this cell. They're drawing it more like a monocyte, maybe. So the viral proteins are getting made in the cytosol. So when that happens, they're floating around the cytosol. Peptide fragments can get picked up in the endoplasmic reticulum, which is where RNA gets translated into protein. So while that's happening, by random chance, some of these get picked up. They're supposed to be picking up your healthy proteins made of the endoplasmic reticulum, and by accident, they'll pick up a viral one, and they'll put it on the cell surface. So now there's like a flag on the outside of your cell saying, I found something suspicious. Okay. MHC class 2, so here's a big difference. This could be any cell. This doesn't have to be an immune cell. So the previous couple slides, it's stated that almost every cell, aside from your red blood cells and platelets, which are just fragments of cells, have MHC class 1. So any skin in your body has this. MHC class 2 is different. It's present only on antigen-presenting cells. So here's the list. Macrophages, microglia in the brain. There's astrocytes too, dendritic cells, and B cells. So they're deriving, most of the time, this is from pathogens taken up from outside, so meaning that they were eaten. And this is a bit of a larger one, 13 to 25 amino acids. So if you ever wonder, I'm putting together a video on Lyme disease right now, like how you could get a tick bite and then become allergic to meat. Like, why is there some crossover? How can you have a viral infection and then end up getting type 1 diabetes? Like, these are all really important things in immunology. So there's only 13 to 25 amino acids, which means every once in a while, whatever little combo is here is present in something other than the virus. So you can accidentally get an immune response to something outside of just the pathogen, because this pattern happens to, by horrible, horrible chance, match something else. All right, and the goal of MHC class 2, these are movable cells. Your skin cells can't really traffic anywhere. They can't go to a lymph node. So MHC class 2 is on these movable cells that can traffic to a lymph node and then go talk to T cells. T cells are in the bloodstream, but they need to go to a lymph node to find a larger number of T cells. Okay, so here's the example. They're saying that there's a bacterium on the outside. It gets eaten by the macrophage. It gets brought into the lysosome and broken into little pieces. One of these little pieces gets put on an MHC class 2 because they're found in vesicles, not in the endoplasmic reticulum. So just when you think cell biology was a bunch of memorization, sometimes you find out that it was memorization for a reason. Okay, so now the MHC class 2 is on the surface and it's got a little piece of a bacteria in it. And now this macrophage has to traffic to a lymph node and find a T cell that matches this little red protein. So remember, this is only 13 to 25 amino acids, which is like tiny, tiny, tiny. Another example. So B cells can't really eat. B cells wear their antibodies on the outside. They do decorate themselves and they release them into your bloodstream. B cells is the next lecture, so we're coming back, don't worry. All right, so here, let's say that this red bacteria piece binds to the antibody. They do internalize it. It's not quite like eating. They kind of pull it back in. But either way, it's in a vesicle, not the endoplasmic reticulum. So here it's in the vesicle, it's getting loaded into MHC class II and put on the surface. Really similar to a macrophage, but there's no like proper phagocytosis. They bind to the antibodies instead. Okay, there's a lot going on here. This internal signaling bit, I would not go over it all right now. It's really for an advanced class. I just wanted you to see that the T cell receptor has these like curved pieces up top. These curved pieces depend on the genes in the T cell. They're arranged randomly throughout your life, and they're meant to have a wide variety of these shapes to bind any piece, any pathogen you ever encounter. So there's an alpha and a beta group here in the T cell receptor. That's all I want you to know right now. So they're found on the T cell surface, and they're the match for the MHC. I might skip ahead just a second. They're the match. So the T cell receptor's in blue, and here we've got MHC class I. I can tell because there's only like one structural protein going through the membrane. But this is it, they match. This is the most important part of immunology. Okay, so the T cell receptor is found on the T cell surface. It recognizes an antigen presented in MHC class II on other immune cells, ones listed before, macrophages, dendritic cells, B cells, microglia. Most T cell receptors have an alpha and beta chain. That was the one thing I pointed out previously. They do also have CD3 molecules as a complex. That doesn't matter unless you actively study the immune system. When I was studying T cells, I often used CD3 as a marker to figure out how many T cells had entered a brain infection or a foot infection, something like that. So the T cell receptor has a constant and variable region, like the B cell receptor, which is coming up next lecture. The genes are called RAGs. It stands for recombination. So these are recombination genes, RAG1 and RAG2. There are animal models to study T cell diseases where they remove RAG1 and RAG2, and it mostly gets rid of B and T cells, and then the rodents are immunocompromised. We'll talk about what VDJ recombination means. Not right now. That's more of a B cell thing. But CD3 requires a friend, a co-receptor to activate. So the two main flavors, types of T cells are CD4 or CD8. And each of these reacts to only one MHC. They're going to either do MHC class II for CD4. Oh, 1 second. All right, I'm back, students. Okay, so where do we leave off? We've got the co-receptors, CD4 or CD8, to activate T cells. If the matching doesn't make sense, we are going to go over that repeatedly, and it's going to come back to what happens in the thymus. Here we go. All right, so this is so important. If you get this, you'll understand a lot of diseases and allergy. There is going to be a separate lecture on allergy and cancer. Okay, so here's the T cell. It's at the top, and the antigen presenting cell is at the bottom. Hopefully you have a few examples in your mind. So this is this over here. This results in an activated T cell. This does not result in an activated T cell, and neither does this. So an antigen presenting cell seeking a T cell doesn't immediately find their match, their soulmate, whatever. It takes a long time. They might go through hundreds or maybe even thousands of T cells to find their match. So here, why didn't this match work? It's the wrong MHC. They have like this curved area to show you that it needs to bind to a curved MHC, even though the little red antigen would fit perfectly. Over here, we have the curved MHC, but this peptide has like a flat top. It doesn't, have the point like the other one. So this one doesn't work because of the peptide. So peptide didn't match here. MHC didn't match here. So it's got to be perfect. Everything has to match up, which is why you have so many millions of T cells for your immune system to be able to seek out a match for anything you find. There are actually a lot of immunology memes, like way more than I thought there would be, but this one's cute. You got the T cell, you got the dendritic cell, handing over the peptide to the MHC, and now the T cell is activated and looks very happy. Okay, so let me move my face out of the way. Types of T cells. If you pick up an immunology book, they go back and forth between the descriptive name and then like the shorter name. So the descriptive name is a helper T cell, CD4, and then there's cytotoxic, CD8. And the descriptive names actually tell you what they're going to do. There's also memory T cells, regulatory T cells. We'll come back to that when we talk about autoimmune disease. Natural killer T cells and gamma delta T cells. But these two together are like less than 5%. Less than 5% of T cells. So they're not going to fit into this lecture. It's more of an advanced concept. 95% of them are helper or cytotoxic. So what the heck does that mean? Helper. It means that they really just release cytokines. They're not going to directly kill anything. These, however, these can directly kill cancer cells and virally infected cells. So here's the helpers. They are basically like macrophage cheerleaders. They release a ton of cytokines. They can destroy intracellular bacteria and indirectly kill through B cell and macrophage activation. So there are different types. There are subtypes of CD4 T cells. The two first discovered and best studied are Th1 and Th2. The type of cytokine, if you recall back to the cytokine lecture, the type of cytokine that they make is interferon gamma, and that's for like viral infections and bacterial infections, and they help those macrophages do the intracellular processes that kill bacteria, like hydrogen peroxide and reactive oxygen species and all that. They help them do it better. Th2 is actually for allergy and parasite. So yes, there's an allergy hypothesis that rests completely between Th1 and Th2 T cells. And I promise that is an upcoming lecture. Okay, so let's visualize it. You've got your macrophage here. Your macrophage has an MHC class 2, which binds to CD4 helper T cell. So this is this process. This T cell is going to activate the macrophage, and the macrophage will be able to defeat the internal bacteria. What else could happen? A helper T cell could find a B cell that has not been activated. So for example, if this is the first time you've encountered this bacteria, you don't have any antibodies to it. You have, like, aside from complement, no protection in your bloodstream. So the helper T cell comes along to the B cell, which is the one cell in your body that makes antibodies, activates it, and then it would turn into a plasma cell, which is just an activated B cell that cranks out lots of antibodies. So then it would be like, okay, boom, antibody against bacteria, antibody against bacteria, but just that tiny little piece. So it could be against the outside of the bacteria, the flagellin, it could be to like RNA or DNA, or even to a bacterial toxin. So this is the kind of thing that you would receive to combat like a tetanus toxin. Okay. CD8 T cells. These are called cytotoxic. They interact with MHC class 1. So that could be present on any cell. And the reason that's important is because cancer and viral infections can happen on any cell. So these cells, which MHC class 1, they kind of stay put, and then they just hope that a CD8 T cell comes along. All right, so they release cytotoxins like perforin and granzyme. You've heard of these with neutrophils. They also can induce cell death. cell death is called apoptosis. I called it apoptosis in grad school, and one of my mentors was like, no, it's apoptosis. Okay. So the reason that they can kill cells is through something called Fas ligand. They just sort of like swips, that's funny. Switches. Did I say that? Flips the switch. That's what sorry. Flips the switch from like alive to dead. And we're coming back to that in the cancer lecture. All right, so how does it work? This is a CD8. I wish they would draw, like, these diagrams are good, but I wish they would just draw the names too. So in purple, for some reason CD8s are often drawn in purple. All right. So the CD8s here, it's talking to whatever, a cancer cell, a virally infected cell. MHC class 1 is here. It binds to the purple T cell receptor, and then it gives it the signal to die, probably through Fas ligand. Okay. So this is another one. I feel like if there's just a few figures that you want to focus on, it's the matching between the T cell receptor and the MHC class 1 or 2. And then this one, this is a good summary slide. Chips and crackers. Here we go. All right. So if you want to find a protein, like where did it come from? So if it came from the cytosol, it's going to be on an MHC class 1. If it came from a vesicle, it's going to be on an MHC class 2. So that can already tell you where it's going to be loaded. Now, what T cell would an MHC class 1 bind? CD8. What T cell would an MHC class 2 bind? CD4. And then this is the result. Cell death for the cytotoxic CD8, and then activation of either B cells or like macrophages. Okay. This is the third concept. How do we even get T cells? They don't mature in the bone. They have to traffic from our bone all the way up to the thymus. So hopefully you can remember if I asked you, are they myeloid progenitors or lymphoid progenitors? Okay, they're lymphoid progenitors, yep. So they're going to traffic to the thymus, and then they have 2 tests. The reason we have these tests in our body is to prevent uncontrolled autoimmune diseases. I'll tell you why. And I usually joke with my students, only 1% of T cells survive, so you're definitely going to do better than that in my class. Sorry, bad teacher joke. Okay, positive selection is that T cells can communicate with your other cells. This is a check to make sure that the T cell can identify either MHC class 1 or MHC class 2. Otherwise, they won't work. Negative selection is getting rid of T cells that react to self. So if the T cell works, but then it really wants to bind to your proteins and your tissue, that is setting yourself up for autoimmune disease. So they get removed or occasionally reprogrammed. So let's take a look. Let's pretend that we took out your thymus, or that we're looking at your thymus, and there's like a little thin slice of it. Okay. So when T cells first get into your thymus, they don't express any classic T cell receptors yet. So they're called double negative because they don't have CD4 or CD8 expressed. So then they're going to interact with some of the cells. They're often called epithelial cells, thymic epithelial cells. All right, so they're interacting here, and now they start to express both CD4 and CD8. So it goes from nothing, double negative, to double positive, both. And then they make a choice. And the choice is down here. If they, and this is chance, so much of the immune system is chance. If they by chance encounter a dendritic cell in the thymus that expresses MHC class 1, they choose to become a CD8 T cell. If they happen to run into a dendritic cell and it's expressing MHC class 2, they turn into CD4 T cells. All right, so let's dive into that a little more. Positive selection is when double positive They call them thymocytes because they're in the thymus. So double positive CD4 and CD8 T cells are presented with self-antigen. If they interact with MHC class 1 or 2 too strongly or too weakly, they could be in trouble. So they're actually, this step also is going for like the perfect work handshake, not too firm, not too weak, just like the perfect middle ground handshake. So they get a survival signal if they interact correctly. So MHC class 2 turns into a CD4 T cell. If the T cells bind to MHC class 1, they turn into CD8, just like I mentioned before. Okay. So this is a serious discussion question. What would happen without MHC class 1 in the body? So you would never get CD8 T cells. And a person without CD8 T cells would be more likely to get serious viral infections. Like every time you get a virus, it would be worse than a person who had it. You might be hospitalized more, things like that. So also they'd be more prone to cancer. Cancer could probably get to a further stage than it would have without them. Okay. So without the thymus, without MHC class 1 in the thymus, no T cells. Without MHC class 1 in the body, like let's pretend you still do have CD8 T cells. The CD8 T cells would have nothing to interact with. So they would be kind of useless. All right, so let's take a look. If there's normal MHC class 2 expression, you can get both CD8 and CD4 T cells. If for some reason you're a very rare person who has a defective MHC class 2, you would only get CD8 T cells. You get no CD4. So that's how important the connection is between the T cell receptor and the MHC class 1 or 2 receptors. All right, I think this is more or less the last concept, the last big concept. So here's negative selection. This is a dendritic cell. They're drawing it like a star. It's in the thymus. They're coloring it yellow. And this is an important step because after they choose to be a CD8 or a CD4 T cell, they have one more check to make sure they're not going to react to self. So these special cells, these dendritic cells, are expressing your healthy proteins. So there should just be, again, that perfect handshake, just some moderate binding, and then they get this happy signal to live. If this binds too tightly, like a really rough handshake, they know that this T cell is probably trouble down the line and might cause you an autoimmune disease. So they actually give it the apoptotic signal to die, which is actually kind of similar to the CD8 T cells, offing viral and cancer-infected cells. All right, so here's the write-up. Thymic epithelial cells express self-antigen, just meaning proteins normally found in your body. T cells that bind too strongly are signaled for self-destruction. Every once in a while, though, you get a survivor. It's very interesting that your body comes up with this program. So some strong self-binding T cells actually get signaled to become T regulatory cells. So the job of a T regulatory cell is to roam around, shutting off other rogue T cells. So they, yeah, I don't know if it's like, I'm thinking of a spy analogy, but yeah, they get flipped. Okay, so one of the other concepts I told you earlier, dadgummit, can't put this in any helpful place, pardon me. Okay, so this says up here, T cell memory formation, 0 days after antigen exposure, all the way to 3 weeks after. This is why every once in a while, somebody tells me, like, I got the flu vaccine and then I got the flu 3 days later. I'm like, I'm so sorry that happened to you, but you can't, you needed to wait 2 to 3 weeks to be fully covered. Now, this is why. So 0 days after antigen exposure, be it virus, bacteria, whatever, the antigen presenting cells need to find the T cells. They're called naive if they've never been expanded before. Clonal expansion is when one single T cell turns into thousands, tens of thousands of T cells. That's why if you've ever had like a bad infection and you can feel your lymph nodes here, and if they ever get swollen, that's your B and T cells making thousands of clones of themselves, and then your lymph nodes like swell up, they get bigger. Okay. There might be some differentiation, and then ideally about 10 days afterwards, most of the virus and bacteria are gone, ideally. So then you actually don't need tens of thousands of T cells. So what happens is there's a contraction phase, and only about 10% of the T cells survive and turn into memory cells, so that the next time you get sick, this happens as you are exposed to it without vaccination, but it also happens with vaccination. The surviving memory cells retain that, and then the next time you get a cold, they fight back much faster. All right, so T memory cells are long-lived. They can live up to your entire lifetime. It's not the same cell, but like their clones can live up to an entire lifetime. Sometimes they only live a year. Sometimes they live 10 years. And the exact reason behind it is unknown and is a really important and hot area of research. Is, why is a flu vaccine good for a year versus your measles, mumps, and rubella is good for your whole life? Like, why? We don't know yet. If you wanted to study them, you can look for CD45RO. This is something I've actually done in bacterial brain infections in a rodent model, and you can try to find these memory cells. They're able to self-renew, which is why they can live longer than other types of T cells. And they quickly expand to combat a second encounter with the antigen. All right, so this is what it looks like. I'm going to move this. I'm so sorry it's getting in the way. All right, so the primary response, you still need this innate immune cell to pick up an antigen, break it down, put it on an MHC, take it. So they're doing CD8. So which MHC is it, 1 or 2? 1? Yeah, good. MHC class 1 takes the T cell and they expand, make thousands of copies, and then go back to wherever the primary response was. Skin, whatever, liver, heart, wherever it is. So the secondary response, you move this. So you actually have these blue memory cells now. So you still do need the innate immune cells every time. They're very important. The innate immune cell has to traffic over here, but then the memory cells make more copies. So you have A, you have more than before, and B, this is faster than before. Okay. All right, we're at the conclusions. So the next lecture is B cells, and then I think we'll do like cancer and allergy. And then I'll try to post something in my original format with the drawing. All right, so MHC class 1 identifies self on all nucleated cells. So remember, red blood cells don't have that. MHC class 2 binds the T cell receptor and helps release cytokines. CD4 is helper recruiter. CD8 is directly killing, cytotoxic. CD4, CD8 interact with different receptors and have different responses. Positive selection is that a moderate binding of T cells gets the survival signal. Negative selection is that strong binding of T cells are removed to reduce our chances of getting an autoimmune disease. So if you connect this lecture to, I think, the second lecture, where I went over the primary and secondary lymphoid tissues, you might remember what happens in T cells. Around 13, around adolescence, your thymus starts to shrink. So what do you think is happening when your thymus is shrinking? You're maybe not getting the best positive and negative selection, so you may end up with an autoimmune disease. Okay, all right, that's it for today. Hope you have a nice weekend.